Good morning, students. Today I'll be talking to you about a new macromolecules which plays a very significant and important role in most of the cellular activities of the cell, and that macromolecule is called protein. Now the term protein is derived from the Greek word which is proteos, means it is holding the first place. Protein occurs in every part of the cell and it constitutes 50% of the cellular dry weight. Now proteins are the most abundant organic molecule of the living system. Now if we see the different protein molecules, the most commonly found proteins in the animal system is the collagen. Collagen is the most abundant protein in the animal world, whereas ribosco, ribulose biphosphate carboxylase oxygenase, this is one of the protein molecule, enzyme protein, which is available plenty in the plant system. So if we see the function of a protein molecule, we can broadly group the function of this protein into two basic division. One is the static or the structural function, another is the dynamic function. Now as far as the structural function is concerned, the name itself indicates that it gives the structural and the strength of the body. Collagen, elastin, etc. are the protein molecules which are found in bone matrix and alpha keratin in the epidermal tissues of the cell. Whereas the dynamic function, it is the protein that which are diversified in nature and they play different catalytic role like enzymes. Enzymes are the biocatalysts and they play different catalytic role in the cell. Similar to that, hormones, immunoglobulin, blood clotting factors, membrane receptor, storage lipid, etc. are coming under these categories. Now in my earlier lecture, I have told you that there are certain molecules which constitute this macromolecular structure, and if we see that carbon, hydrogen, oxygen, nitrogen and sulfur are the basic constituent of this protein molecule. If we see the percentage of carbon, it varies from 50 to 55%, whereas nitrogen 6 to 7%, oxygen 19 to 24%, nitrogen 13 to 19% and sulfur 0 to 4%. That means if we see the major constituent, we can find that the carbon, hydrogen, oxygen, nitrogen and sulfur are the basic constituent of the protein molecule. Besides this above molecules, protein contains some elements such as phosphorus, ferrous, copper, iodine, magnesium, manganese, zinc, etc. within it. The content of this nitrogen, an essential component of the protein, varies on an average to 16%. Now this is the basic constituent of the molecules which constitute the protein. Now when we are talking about this amino acids are the basic building block of protein molecule. Now proteins are the macromolecules which are polymers in nature, polymeric in nature, and the basic constituent of these polymers are the alpha amino acid. There are more than 300 amino acids which are there in this nature, but only 20 amino acids are considered to be the standard amino acid, and with its permutation and combinations protein structure is made. Amino acid consists of two fundamental group. That means, if we see the structure of amino acid, we can find that this amino acid has got one carboxyl end, amino acid has got one amino end, and one amino acid is different from the another amino acid with its R group. So, this is the side chain, and this side chain varies from one amino acid to the another amino acid. So, this is the fundamental basic structure of any amino acid, and as this is the first carbon, then this functional carbon, this is the alpha carbon, and this is the basic structure of amino acid. So, if we see the structure, one hydrogen is present, one carboxyl end is there, carboxylic acid and COOH group is there, one amino group is there, and the side chain R group is present in the basic core structure of amino acid. Now, if we divide this 20 standard amino acid based on its characteristics or based on the properties of this R group, it can be categorized into 4 different divisions. One is called the non-polar or the hydrophobic amino acid. Second group is the polar or hydrophilic amino acid. Third is the acidic amino acid, and fourth group is the basic amino acid. Now, whatever is there, the 20 standard amino acids has been divided into these 4 different categories. So, this 20 standard amino acid has been divided into 4 different categories. Some are non-polar, some are polar, some are acidic, and some are basic in nature. Now, let us come to the non-polar amino acid. Alanine, proline, valine, phenylalanine, tyrosine, tryptophan, methionine, isoleucine and leucine are the non-polar or the hydrophobic amino acid. What is the basic nature of this type of amino acid? They are insoluble, mostly or sparingly soluble in water. Now, these are the R group. So, when I am talking to you, this is the R group and this is the core moieties of the amino acid. These core moieties are same in most of the amino acids. Now, when we are coming to the polar R group, then we can find that serine, threonine, cysteine, glycine, asparagine, glutamine are the polar amino acid, where glycine is the amino acid which is placed on the transition phase of polar and non-polar amino acid. Now, this is the only amino acid where this carbon is not an asymmetric carbon atom. That means, here this R group is also another hydrogen H. So, as this is H, this glycine is not having this chiral carbon within it. When we are coming to the basic amino acid, it is otherwise known as positively charged amino acid, and here we can find that in case of lysine, here that additional amino containing groups are there. Arginine has got gonadinium group and histidine has got imidazolium group within it, and here they have this additional amino containing group within it, and that is the reason why it is called the basic amino acid. Whereas the negatively charged amino acids are the aspartic acid and the glutamic acid. Now here I would like to tell you that amino acid, if we see the core structure of this amino acid, as I have already discussed, that it has got the carboxyl end and another is the amino end. So besides this carboxylic group and the within the core structure of this particular amino acid, additional COOH group is present in both these amino acid, that aspartic acid and glutamic acid, resulting the nature of this amino acid acidic in nature. So these are the broad classification of this amino acid. Amino acid can also be classified based on this aromatic ring. So some groups can be also once again taken which contain the aromatic amino acid, such as this phenylalanine, tyrosine, tryptophan and so on. So based on its structure, the 20 standard amino acid has been classified. Now these amino acids are playing a very important role as far as the mechanism, the cell functionality is concerned, and that is the reason this amino acids has been classified or symbolized with either 3-letter digit or with 1-letter digit. Say for example, now glycine, if we consider glycine as one of the amino acid, it can be denoted by GLY, that is the 3-letter symbol, and in single letter it is G. Alanine is A or ALA and A, valine is VAL or V, leucine is LEU or L. So in this way, these 20 standard amino acids are classified with the 3-letter and single letter symbol. The significance of this single letter and 3-digit symbol you will be understanding gradually when we will be going a little bit in depth of, and you will be learning the role of other macromolecular structure and functionality of that particular macromolecule in the cell system. So these are the 3-letter and single letter symbol of amino acid. Now as I have told you, this is the basic ionic form of amino acid. This is the carboxyl end and this is the amino end and this R group. So as I have already told you that R group of each 20 standard amino acids varies from each other, and which makes one amino acid different from another amino acid. Now if we go for this classification of amino acid, then amino acids are classified based on the structure and the chemical nature, that is the nutritional requirement and the metabolic activities of any living cell. Now based on the nutritional requirement, then this amino acids has been classified into 2 types. One is called the essential amino acid, another is the non-essential amino acid. Now what is essential amino acid? These essential amino acids which cannot be synthesized by our body system. That means for the supplementation of these amino acids, we have to supplement these amino acids through diet. And what are those amino acids which cannot be synthesized by the body? They are arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine and tryptophan. So these are the amino acids which cannot be synthesized within the body system, and that is the reason these amino acids are replaced, or it is that demand is met through diet or food. Arginine and histidines are the amino acids which are synthesized by the adult, but it cannot be, it is not present in the growing children. In case of non-essential amino acid, that means those amino acids which can be synthesized by the body system itself, that means glycine, alanine, serine, cysteine, aspartic acid, asparagine, glutamic acid, glutamine, tyrosine and proline. These are the amino acid. Body can synthesize these amino acid by itself. So, supplementation of these amino acids are not that essential. Now, when we are coming to the properties of this amino acid. Now, amino acid can be denoted by a proton, or it can donate proton or accept proton due to the presence of the COOH group and amino group present within it. Zwitterionic form, it is a hybrid molecule containing a positively charged as well as a negatively charged ionic group within it. That means, if we see this structure, it has got positive charge as well as negative charge. Resulting charge in the zwitterionic condition is 0. That means, no net charge is present on the surface of the amino acid. Isoelectric point, it is isoelectric pH is that pH where a molecule exists as a zwitterionic condition. That means, no net charge is there. So, positive charge is equal to the negative charge. That means, when we are passing this protein molecule or this amino acid in an electrical field, there will be no movement because of the net charge, as it is 0 or neutral. Now, this amino acid can be seen by the Fischer projection. All amino acids except glycine, I have already mentioned that in glycine the R group is simply H, and that is the reason it is not the chiral molecule. Amino acids have stereoisomer, and in biological system only L form of amino acids are used in the protein molecule and not the D form. This is the L form and this is the D form. So, this L form of amino acid is present in the protein molecule, but in D form this is found in the larvae and pupae of the insect. So, these are not playing any role as far as the protein is concerned. Now, when we are talking about the D and L form of amino acid, we can find that every amino acid has a similar basic structure. That means, one amino terminal, another carboxy terminals are there, except for glycine where R is H. All amino acids have at least one asymmetric carbon atom and exist as two stereoisomer, that is either the D form or the L form. This is the D form or the L form. So, why? This is the chiral carbon and it is the amino group is present here, and in the D form the amino group is present on the right hand side. So, this is the D form and L form of this amino acid. So, stereoisomeric character is one of the very important properties which determines the L form or D form of this amino acid. Now, amino acid, when it is acting as acid in solution more basic than the pi, that pi, what is the pi? It is the isoelectric point where there is no net charge which is present on this amino acid. The aminos NH3 plus in the amino acid donate a proton to this and it forms this ionic structure. So, this is the zwitterionic at the neutral pH pi, and here under the the basic condition where hydroxyl group are there. So, in higher pH, this Pi is having this, it can donate a proton to this and making this structure NH2, CH2 and COO minus. Similarly, when the solution is more acidic than the Pi, the COO minus, that carboxyl group in the amino acid, accept a proton, and this is the structure of this, and it becomes NH3, CH2 and COOH, and this is the characteristics or the behavioral properties of amino acid. Now, coming to the little complex structure of this amino acid. I have already mentioned you that these amino acids are the basic building block of protein molecule. That means this amino acid constitutes the protein molecule. So, how it is? That means it is joining together, and when one amino acid is joining with another amino acid, the joining it forms the amide bond. Now, what is that? Now, if we see the structure of one amino acid, now see, this is the structure of one amino acid. This is the R1, this is CH, this is the amino terminal, and this is the carboxy terminal. Now, when one amino acid is joining with the another amino acid, that means I have already mentioned you that amino acid has got two terminal, one is the amino end, another is the carboxyl end. So, one carboxyl end of one amino acid, when it joins with the amino terminal of the second amino acid, they form the amide bond. Now, when COOH is coming in contact with this NH2, they release a molecule of water from this, and this bond is called the amide bond. Now, in this way, when it forms a chain like structure, it is called the peptide bonding. So, peptide bond is the amide bond between the carboxyl group of one amino acid. This is the first amino acid, this is the second amino acid, this is the third amino acid, and this is the fourth amino acid. So, when these amino acids are joining together and making a chain like structure, then it is called the peptide bonding. Now, classification, and they form the protein structure. Now, when we are coming to this protein, now let us come to this classification of protein. Now, based on the amino acid composition, structure, shape and solubility properties of the protein are broadly classified into three major groups. First group is called the simple protein. They are composed of only the amino acid residues. It consists of the globular and the fibrous protein. Example is albumin, albumin, globulin, histone, collagen, etc. Now, when we are coming to this globular protein, it is usually water soluble, compact and roughly spherical in nature, hydrophobic interior and hydrophilic surface. Now, globular protein includes the enzyme carrier and the regulatory protein. That means, if we see the nature of this amino acid, I told based on the characteristics of these amino acids, that means hydrophilic amino acids are present on the surface of the protein, whereas hydrophobic moieties, because hydrophobic are water hating, water is not a very suitable environment for them, they try to bury it inside, which is covered by the hydrophilic moieties. So, this type of combinations, it forms the protein structure and it is forming the globular protein. When we are coming to this fibrous protein, it provides the mechanical support, often assembled in a large cable or thread like structure. Alpha keratin, which is a major component of hairs, nails, etc., are the example of alpha keratin which are insoluble in water. Collagen are the major component of the tendon, skin, bones, teeth, etc., where these collagen proteins are there. So, you can understand that all these biomolecules which we are carrying are insoluble protein, that fibrous protein molecule. The conjugated proteins are, besides this amino acid, this protein contain non-protein moieties along with it, which is called the prosthetic group. Nucleoprotein, glycoprotein are some of the example of this conjugated protein. Now, in case of nucleoprotein, nucleic acid is there as the prosthetic group for the functionality of that particular protein, whereas carbohydrate moieties are present sometimes as a prosthetic group in the protein molecule for its functionality, like mucin, which is present in the saliva. Sometimes and it is categorized as the derived protein. These are the denatured or degraded products of the simple and conjugated protein. Examples are the coagulated albumin, that is the egg white, fibrin, which is formed from the fibrinogen, and proteases, etc., are the example of the derived protein. Now, when I have given you enough example of this protein. Now, if we see the organization level of this protein, then we can categorize the structure of this protein molecule into different groups, which is called the primary structure, secondary structure of the protein, tertiary structure of the protein, and the quaternary structure of the protein molecule. Now, if we see the basis of this structure, primary structure is the amino acid sequencing, and it is the simple amino acid what I have told you, I have shown you here, that one amino acid, one carboxy terminal of this amino acid is joining with the amino terminal of another amino acid, and in this way they are forming a big chain, resulting one amino terminal free and another carboxy terminal free of this chain. When we are coming to the secondary structure, it is the folding into the alpha helix and beta sheet or random coil. That means here, besides this primary structure, there are additional bonding which is called the hydrogen bonding is present. When we are talking about the tertiary structure, here 3D folding of a single polypeptides are there, which may be hydrogen or disulfide bond, electrostatic or hydrophobic interactions are also present within this tertiary structure. And when we are talking about this quaternary structure, it is considered to be the more complex, and here the association of different peptide bonds subunits are there and they are folded within it, and it is having the more complexity and bonding pattern is same as that of the tertiary structure. So, if we see the primary structure, this is the simple structure. Secondary structure, it is trying to form a coil. Then tertiary structure, it is the globular protein and trying to make more complex structure, and quaternary is the most complex structure which is there within the protein molecule. Now, coming to this primary structure, I have already discussed this primary structure. It is the simple peptide bond which is having a very straight chain. When coming to this secondary structure. Secondary structure of protein indicates the arrangement of the peptide chain in a space. Now see, here this is the chain, peptide chain, and here it tries to be a coil like structure. When it is trying to be a coil like structure, additional this amine containing group has got some hydrogen bonding with the carboxy end of another amino acid, and they are trying to form little bit complex structure, and they try to associate the coil within this conformation, and that is this coil shape of this alpha helix is held in a place by hydrogen bond between the amide group and the carbonyl group of the amino acid. Now if this is the black is the carbon, red is the oxygen, nitrogen is the violet, R group is also the dark violet, and pale blue is the hydrogen, then we can find that say this bonding has got this carboxyl, this oxygen is getting bonded with hydrogen and they are associated with each other, and this amino containing group has got linked with the carboxyl end with the amide bonding, and this way they are associating, they are making the link. When we are talking about the secondary structure, it is called the beta pleated sheet. Now here it holds the protein in a parallel arrangement. So these are the peptide linkage and they are just trying to hold themselves in the parallel arrangement, and it looks like a matrix like structure. It has a R group that extend above and below the sheet. This above and below the sheet, it is a typical fibrous protein such as silk. So this is one of the example of the beta pleated protein. This secondary structure, we can also get the triple helix. It consists of 3 alpha helical chain. It contains a large amount of glycine, proline, hydroxyproline. This is a derivative product of proline, hydroxylysine that contain the OH group for hydrogen bonding. It is found in collagen, connective tissue, skin, tendon and the cartilage of the body. Now this is the tertiary structure of the protein molecule. When we are talking about the tertiary structure, if it gives the specific or overall shape to a protein that involves the interaction and cross links between the different part of the peptide chain. Hydrophobic and hydrophilic interactions are there, salt bridges are there, hydrogen bonding and disulfide bonding, that is the cysteine molecule. When cysteine is the amino acid which contains sulfur. Methionine is also another example of sulfur containing amino acid. So disulfide bridges, when such type of amino acids are present, they form the disulfide bridges. So when we are talking about this tertiary structure, this secondary structure, when it is getting bent or folded into a more complex 3D structure, we call it as a tertiary structure. Here, besides this peptide and the hydrogen bonding, we can expect all possible of other bonding like ionic, disulfide, van der Waals, hydrophobic and so on. Every reactions, every bonding are possible because it is more and more folded together to form this structure. Now here hydrophobic, ionic, hydrogen bonds, disulfide bond etc. are present for proper aggregation of the individual protein molecule, and it forms the tertiary structure. Now when we are talking about this type of chain, they are called the peptide chain. Now, in this tertiary structure, it is the peptide chain which is folded, bent, and they form the complex molecule. Now, in case of quaternary structure, it is more and more complex, I have already told. Now, when such peptide, more than one peptide chains are getting associated with each other. Now, say for example, hemoglobin is one of the example of quaternary structure of the protein. Here 2 beta chain and 2 alpha chains are associated with each other, and they are forming the very complex structure. And when we are talking about this association, obviously whatever bonding, whatever complexities are there, so all the peptides are linked together and they form a jumbled like structure, and here this type of structure, these complexities are found in quaternary structure of the protein. So, quaternary structure is the interaction between the adjacent polypeptide that make up the single protein, that is hemoglobin, collagen, etc. are the example of the quaternary structure of the protein molecule. So, I wanted to give you some idea about the structure and function of different protein. Now, how to quantify, how to know that what is the protein concentration in the particular environment. Now, we can measure directly or indirectly. Now, Gildal method is one of the technique through which we can quantify the total organic nitrogen present in the system, and then when we are multiplying it with 6.25, we are getting the approximate quantification of this protein molecule which is present in that particular solution. When this is the dye binding method is also another technique where we are using some specific dye. Suppose Coercive brilliant blue is one of the dye which is otherwise known as Bradford method of protein quantification, which has got a direct link with this protein. More the concentration of protein, more will be the binding of this dye to that particular protein, more will be the color intensity, and through this quantification standard quantification method, we can determine the protein content of that unknown solution. Similarly, biurate method, Lowry's methods are some of the standard quantification method of protein estimation or protein concentration determination. Ultraviolet method is also one of the technique where without using any dye, any reaction medium, if we are taking the protein solution, colorless protein solution, and if we are measuring the optical density at 280 nanometer, then we can find we can quantify that what is the protein concentration present in that particular solution. It is worth mentioning that 260 nanometer is the optical density for nucleic acid and 280 nanometer is the wavelength where we can quantify the protein solution in any solution. Unknown solution, colorless protein, if it is present, we can quantify that. Now, coming to the another important aspect of the protein molecule. Now, enzyme is one of the important biocatalyst which is present in every living system. It is worth mentioning that all enzymes are protein, but all proteins are not enzymes. Now, here we can find that enzymes are the biocatalyst which synthesize by the living cell. They are proteinaceous in nature. Some exception is there except ribozyme, that is the one enzyme which is non-proteinaceous in nature, colloidal, thermostable and specific in action. Kuhne used the word enzyme to indicate the catalysis of the biological system, and Buchner isolated the zymase enzyme from the yeast. Now, when we see the chemical nature of this enzyme, we can find that all the enzymes are invariably proteinaceous in nature. I have already told that except the ribozyme. That functional unit of enzyme is known as the holoenzyme. That means the functional unit is called holoenzyme. Holoenzyme is made up of apoenzyme. What is apoenzyme? This is the proteinaceous part of the enzyme, and the coenzyme is the non-proteinaceous part, which is, it may be the organic other part of this body which is not proteinaceous in nature. Besides this, some cofactor like prosthetic group, coenzyme and metal ions are also playing a significant role as far as the functionality or the characteristics of protein is concerned. Now, when we are talking about the prosthetic groups are the group that are tightly bound with the apoenzyme. Apoenzyme means the proteinaceous part of the enzyme. Now, when we are talking about this coenzyme bound transiently to apoenzyme and they are separable by dialysis. Example is vitamin, nicotinamide adenine dinucleotide, that is NAD, contains niacin as the vitamin. Metal ion forms a coordination bond with the side chain of active site concomitantly with the substrate. Zinc is a cofactor for carboxypeptidase, that is the proteolytic enzyme. Now, if we classify the enzymes based on its catalytic behavior, the entire enzyme can be divided into 6 major classes. First group is called the oxidoreductase group of enzyme, second group is the transferase, third group is the hydrolase, fourth group is the lyase, fifth group is the isomerase, sixth group is the ligases. Now, suppose if anyone is talking to you about a particular number, suppose 3.2.1.1, EC 3.2.1.1. What does it mean? EC means enzyme commission. Now, 3 means this is the third group of this enzyme. That means that particular person is talking about the enzyme which belongs to this category of hydrolase. Then it is further classified, this enzyme hydrolase is further classified based on its characteristics and the behavior, and that is the reason this sequence is so important. When someone is talking about 1 point something, that means he is talking about the first group of enzyme, that is the oxidoreductase group of enzyme. Now, let us come to that. When we are talking about the oxidoreductase group of enzyme, the name itself is indicating that different type of oxidation reduction reactions are carried out by this particular enzyme. Most of the intracellular enzymes belong to this oxidoreductase group of enzyme. This enzyme involved in the oxidation reduction reaction. Example is the succinate dehydrogenase, which reduces FAD to FADH2 and oxidizes succinate in TCA cycle. I will be coming to this particular step in some subsequent classes when I will be talking to you about citric acid cycle. So, this is one of the example of this oxidoreductase group of enzyme. Now, as a whole, if any dehydrogenase group of enzyme, if any kinase group of enzyme are there, they belongs to this oxidoreductase group of enzyme. Now, transferase is another group of enzyme. This is the second group of enzyme where this enzyme catalyzes the transfer of the functional group hexokinase. Now, hexokinase is one of the example where glucose is getting converted to glucose 6-phosphate with the help of the enzyme hexokinase. What it is doing? It is transferring that glucose, that phosphate group from to the 6th position of this hexose sugar, and it is called the transferase group of enzyme. Now, when we are coming to this hydrolysis brings the hydrolytic reaction of the various compound. Say for example, the lipases, proteases, amylases, cellulases, etc. are the example of hydrolytic enzyme. Now, they breaks mainly this lipase, they are very specific to this ester bond. Proteases mainly break the peptide bond. Amylases and cellulases mainly breaks the glycosidic linkages of the protein molecule. When we are talking about the liases, enzymes which are specialized in addition to double bond such as C double bond C, C double bond O, C double bond N, etc. and it is also removes the water from this reaction. Say this aconitase group of enzyme. I will be talking to you this group of enzyme when in the subsequent classes. Coming to this isomerase, any type of isomerization reactions are carried out by this group of enzyme. Say for example, racemase. Racemase is one of the example. Phosphohexose isomerase is another example of which belongs to this particular group of enzyme. And ligases, the formation of bond with ATP cleavage, the enzymes catalyzing the synthetic reaction where two molecules are joined together and one ATP is used, that is the DNA ligase. So, these are some of the example of the different classes and function of enzymes. Now, when we are comparing, what is the importance of this biocatalyst? Now, when we are comparing this biocatalyst with any conventional catalyst, then we can find that it works efficiently at higher temperature and higher pressure. In that any chemical catalyst can work under any high temperature and high pressure, but in case of biocatalyst, it cannot, because it cannot cross the physiological condition of the living cell, and living cell cannot work at very high temperature or else it will collapse. So, slow catalytic reactions are observed, but here the biocatalysts are highly specific towards its targeted substrate molecule, and that is the reason the velocity of reaction is much higher compared to any chemical catalyst. Now, say for example, when enzyme catalyzes any reaction at normal body temperature, say carbon dioxide and water in presence of carbonic anhydrase, it is producing by H2CO3. This, in absence of the enzyme, 200 molecules of carbonic acid was formed in an hour. In presence of enzyme carbonic anhydrase, this is the enzyme, when it catalyzed this reaction, 6 lakhs molecules per second was formed within this. You just compare and see how efficient these biocatalysts are, and that is the reason why these biocatalysts are occupying such a significant position as far as the living system is concerned. Reaction rate accelerates by 10 million times. Enzyme brings down the energy barrier and facilitate the faster rates. Now, what is that? Now, a chemical reaction, if we consider any chemical reaction that A is giving the product P. So, any chemical reaction, suppose A is giving the product P, which takes place because of certain fraction of the population of A molecules, which gives the instant process enough energy to attain the activation condition called the transition state. And if we see, this is the transition state of substrate to product formation in absence of enzyme. Now, if we compare the transition state energy barrier without enzyme and with enzyme, then energy barrier with enzyme is very high compared to the presence of catalyst for any product formation. This is the top of the energy barrier and separating the reactant, that is A, to the product, which is P. Now, when we are talking about this, then we are obviously coming to this, that how A can produce this product, this P. There are two ways in which this rate of chemical reactions can be accelerated. One is that by increasing the heat of this reaction, by increasing the temperature to attain this transition state. Another is the addition of catalyst or biological catalyst, which work mainly on the physiological conditions, and it is mainly working and it is converting this substrate or the reactant to the product molecule. Now, as this particular temperature which is there, the transition state, the energy barrier is so high, and in compared to this particular enzyme mediated reaction, the activation energy is so less. Obviously, the energy, that is the energy barrier which is there for facilitating the energy, this reaction is much favorable, and the rate of reaction can be accelerated with the addition of the enzyme, which lower down the activation energy of any process. And this is the reason why these enzymes are so important, and this enzyme, how the specificity is so important. There are several factors which play a very significant role as far as the enzyme reactions are concerned. That is the temperature, pH, substrate concentration, binding of specific chemicals that regulate the activity of the enzymes, and the factor alters the tertiary structure and eventually loses the biological activity of the particular reaction. So, these are some of the factors which plays a very significant role. As I have told you, that proteins are the biological molecules, and sometimes depending upon the nature, it may be heat labile, it may be cold labile. So, depending upon the nature of the protein, the catalytic reactions are designed. Now, if we give some example of this protein, these proteins have a wide range of function which includes their role as enzymes, hormone, antibodies, transporters, muscle fiber, the lens protein of the eyes, feather spider web, rhinoceros horn, milk proteins, antibiotics, mushroom poison, and mirrors of the substances having distinct biological activities. Among these protein products, the enzymes are the most varied and the specialized biochemicals. Virtually all the cellular reactions are catalyzed by the enzyme molecule, and with this, I think I have tried to give you the overview of this protein molecule, how proteins are formed, what are the basic units of this building block of this protein, what are the different characteristics of the amino acids. So, I have and this primary, secondary, tertiary and quaternary structure of the protein molecule. Then I have also tried to give you some idea about these enzymes and how enzyme functions. What are the different parameters which governs the enzyme activity. I think you have got some idea about this protein and its structure. So, in my next class, I will be talking to you about another macromolecules which also play a very significant role in the cellular activities. Thank you very much.